We've come a long way in treating lupus. So go back to the 1940s before we had steroids, and you can see the survival over here on this y-axis. So you start out at 100%, and here's time after diagnosis, 1, 5, and 10 years. So pre-steroids, there's about a 50% 5-year survival. We're going back to the early part of the 1900s. And then steroids come along, and you see patients are doing better. They're living longer, and living better, I'm sure. And then the introduction of immunosuppressives and cytotoxics, and you see yet better survival curves. But what we want is a flat curve from 100% all the way out. We want to see every lupus patient live a long life and a good life. So we need better drugs. And these are the driving forces behind lupus drug development. We need drugs with better efficacy, so they need to work better. And I think in particular for lupus kidney disease, but also for severe extrarenal disease, for those patients who have severe arthritis and severe head-to-toe skin rash, and you could go through the whole list. We need drugs that prevent flares. And on the wish list would be, let's get rid of this disease. So remission induction. So at the same time that we need better efficacy, we need better safety. I mean, I just gave you a partial list of all the toxicities of these drugs. But we'd like to see steroids go away. We would like to see chemotherapy go away. We'd like to see the immunosuppressives go away. But as mentioned at the very beginning, lupus is a tough nut to crack. It's hard to design trials. Why is that? Because of the heterogeneity of manifestations. So you can't go to the FDA and say, well, we have this drug, we think it works pretty well because the patients told us. You have to go to the FDA with data, hardcore data. So how do you design trials? Do you just limit the trial to lupus rash or to lupus arthritis? Or do you take all comers, no matter what manifestation they have? So the heterogeneity of manifestations make trial design very difficult. It not only makes trial design difficult, it makes the endpoints. So one has to figure out what a successful treatment is. So the heterogeneity complicates the entry criteria, the trial design, and the endpoints. And not that we have a lot of medicines that we use in lupus, but patients come into these trials on all different kinds of medicines and all different doses. Some will come in on prednisone, some will come in on Plaquenil, some will come in on both, and then you can introduce the anti-metabolites, Imuran and Cellcept. And it's very difficult to sort out if someone's getting better from background therapy that they're already on or the introduction of an experimental medicine. And we're also wrestling with trial endpoints. I know it should sound easy that we should be able to tell who's better and who's not, but I've been doing this now for probably about 18 years, and we design these trials in groups. I call it the lupus mafia. And you would think after 18 years, the lupus mafia would have figured out how to design a trial. But it is very, very difficult. All right, here's a slide that will put you to sleep. This is a slide to demonstrate the different players in the immune system that are malfunctioning in lupus. And I'm just going to run through this very fast. The point of it is to show you that there are all different pathways in lupus, which means there are different targets that different drug companies can target. So in this particular slide of mine, we have a patient with an increased risk. So we know that there's a genetic risk to lupus. But genetic risk is not alone. There has to be a trigger. So we take a genetically susceptible host, and there's a trigger. So in this particular slide, I have the sun as the trigger. And what does the sun do? The sun beams down, hits the skin, and causes certain cells to die. And those cells release DNA. And the DNA is taken up into a particular cell, plasmacytoid dendritic cell, and a particular receptor called a toll-like receptor. And it causes the cell to become very active. And that active cell starts spewing out all kinds of pro-inflammatory proteins. chief of which is something called interferon alpha. And interferon alpha can then stimulate a whole bunch of other cells, which you see on the left-hand side. And then a particular cell, the T cell, is stimulated, the B cell is stimulated, and B cells make antibodies. But what happens is the B cell and T cell come together and they talk. And they both get very stimulated. And these are just some of the molecules through which they interact. So again, more targets for drug development. Break up those interactions so the B cells and the T cells are not stimulating each other. B cells are further stimulated by certain proteins. One is called APRIL and one is called BLyS. And BLyS happens to be the target for that drug that I mentioned, Benlysta, which was approved a year ago. So if you understand the science in the patient, you can then go forward and develop drugs to stop all this inflammation. And B cells turn into plasma cells, which are little factories that spew out antibodies. And in the case of lupus, anti-DNA antibodies. And anti-DNA antibodies can further stimulate various components of the immune system. Complement, and these little cells off the screen are white blood cells called neutrophils and macrophages. And then the whole thing just keeps recycling. So that's a simplistic view of the pathogenesis of lupus, of how lupus starts. So each of those things I mentioned might be a potential target for drug development. I'm just going to focus for the last few minutes on B cells. B cells or B lymphocytes are cells of the immune system. And what B cells do is they make antibodies. And those antibodies in the case of lupus could be anti-DNA antibodies or anti-SSA antibodies or a whole bunch of different antibodies. It's not good to have those antibodies, especially DNA antibodies. B cells can also talk to other cells like T cells and stimulate them, and then you get that whole cascade going. B cells are dependent upon certain growth factors to do their thing. And one of those factors over here is BLyS. And BLyS stands for B lymphocyte stimulator. So that protein, which we all have in our bodies, though levels are higher in lupus patients, stimulates B cells. So that's not such a good thing for a lupus patient. There are different ways to antagonize the effects of that particular protein. And they're sitting over here. And the one I want to talk about is an antibody. Antibodies are natural, obviously. We all make antibodies, part of our defense system. But they've also been used therapeutically. So companies can now mass produce therapeutic antibodies. And they're highly targeted. So one antibody binds to one particular protein. And in this case, this is Benlysta binding to BLyS. And the consequence of that antibody binding to BLyS is it takes BLyS out of the system. It reduces the BLyS levels, which means that BLyS is no longer present to stimulate this B cell through some of these receptors on the surface. So we all have BLyS. Lupus patients have higher levels of BLyS. You can antagonize BLyS with a therapeutic antibody. BLyS levels then go down, maybe even to 0, which means that that B cell is no longer stimulated because there's no BLyS around. So that's just the principle. So this is the rationale for doing this. BLyS is crucial to B cells. And we've shown, before going into humans, there are animal models. I don't know if you know that there are mouse models of lupus. So they're good to study because you can learn a lot before you start studying humans. And to make a long story short, if you give an antagonist of BLyS to a mouse with lupus, you can make the lupus get better. We also did studies in humans looking at BLyS levels and found that BLyS levels were high. And BLyS levels were predictive of flare. So this led to a whole development program. So when drugs are ready to be studied in people, they'll usually do a small study, a phase 1 study, to learn how the drug is metabolized in the body and is it doing what it's supposed to do. They're not really concentrating on whether it's effective or not. They certainly will concentrate on is it safe or not. But in a phase 1 study, which is usually about 50 to 60 patients. We'll generate data, what we call pharmacokinetics, pharmacodynamics, and safety. And then we move from phase 1 to phase 2, which is a larger study, focusing a little bit more now on whether the drug is effective. So with Benlysta, a phase 1 study was done, and it showed that the drug was safe, and we learned about how it's metabolized, and it did bind to Bliss, and it did affect those B cells. But we really didn't know whether it was effective in reducing disease activity in lupus. So then a phase 2 study was done with 440 patients. And that unfortunately failed to show efficacy of the drug. So the endpoint was not reached. That drug did not work in the 440 patients. But then we start dissecting all the different subsets and doing multiple analyses to see where it failed. And that was very valuable in this particular situation because we actually saw in certain subsets of patients, actually the sicker patients, that it was an effective drug. And also what came out of these analyses, I had mentioned before, you can't go to the FDA and say, I think my drug works. You have to go to the FDA with hardcore data. And we've been wrestling with, well, what is a good endpoint for a study? What came out of that particular study was an index, a responder index. We were able to use certain things, and I'm not going to go into any detail, but these are some of those disease activity instruments that I mentioned. And we could say whether a patient was a responder or a non-responder to the drug. And though it sounds very simple, it really was a major breakthrough because we never used a composite index in lupus. They're very commonly used in osteoarthritis studies and in rheumatoid arthritis studies. So here's some of the data. We'll just spend a little time on this. The drug went forward into the next phase of studies, phase 3. And phase 3 is the real thing. Phase 3, the FDA requires 2 studies to be done. If you're successful in both studies, it's very likely that you'll get your drug approved. Barring any safety problems. This was with Benlysta. There were 2 major studies done, each enrolling about 800 patients. One was done in the US and Western Europe, and the other was done in South America, Asia, and other parts of the world. And you see these little bar graphs. One study, Bliss 52 on the left, and the US study, Bliss 76 on the right. And on this y-axis, the percentage of patients achieving that response. And you have placebo, so in a typical study, patients will be assigned to get the drug, and maybe different doses of the drug being looked at, and some might get a placebo, and a placebo is an inactive therapy. Now, we're not being cruel in giving someone placebo because all of these patients are receiving therapy already. So the typical patient in this study was on steroids and maybe an immunosuppressive. So about 2/3 of the patients were on 2 or more background therapies, and they received either a placebo or the experimental drug at 2 different doses. These were patients who still had disease activity despite being on those background therapies. And you can see the response rates. So 43% of the people who got placebo actually responded using that definition, but higher numbers receiving Benlysta responded. And the same kind of pattern in the other study. So 2 positive phase 3 studies leading to an FDA approval in March of 2011. So I don't know how much you know about Benlysta. We're all learning about Benlysta. The rheumatology community has the following questions, so do the patients. Who should be treated with this biologic? And we're learning our way. So basically patients who still have disease activity despite some of the medicines that I mentioned earlier on. Which manifestations respond the best? People are always asking about that. Well, a good chunk of the patients in this study had arthritis or rash. And those are 2 manifestations that actually did better on drug than placebo. When is a response expected? It's not a quick to work drug. It takes many months to see an effect, so you have to be a little bit patient. It's not like taking steroids and seeing an effect 2 days later. And then other questions that people have, when should failure be declared? And each physician is going to give you a different answer. And if successful, how long should it be continued? And another question is that responder index that I showed you. A lot of people are confused by what that really translates into. And there's a lot of literature coming out about the clinical significance of that lupus responder index. So the lessons learned from lupus clinical trials. There have been a lot of trials. There have been a lot of drugs developed, or at least attempts to develop drugs in lupus, a lot of trials. Only 3 have been successful. So I showed you 2, and there was another one. So what we've learned about doing lupus clinical trials to develop new drugs is that it's a very humbling experience. We had some surprising failures. Rituxan. It made a lot of sense to use Rituxan or rituximab in lupus. So we did 2 trials, one in kidney disease and one in extrarenal disease. Both were failures. And is it because the drug doesn't work in lupus? Well, that's hard to convince rheumatologists because we're using that drug for certain manifestations. And in other countries, especially England, they rely on it a lot. Or was it the trial design? And as I mentioned, it's very difficult to design trials. So a bit of a breakthrough was that responder index, and there are variations on that theme that other sponsors are using. So that was a surprising failure. We thought it would be a shoo-in, just do 2 studies and then we'll have another drug for lupus. It didn't happen that way. There have been some successful failures. CellCept. The FDA has a policy that when you do trials, you have to show superiority of your experimental drug to an already approved drug. It's just there are very few approved drugs in lupus. So this study was designed to show that CellCept was equivalent to Cytoxan. As I mentioned, we want to get rid of Cytoxan for kidney disease. We thought CellCept would be better, but the rule was that to get an approval, the CellCept had to be better than Cytoxan. Anyway, in the study it showed equivalence between the 2 types of drugs. And I call it a successful failure because it told us what we thought we knew, that is that CellCept is a good drug, but it unfortunately did not lead to an approval in lupus. But fortunately, the drug is approved for other indications. So with a kind insurance company, we can get CellCept for our patients with lupus, kidney disease, and other manifestations of lupus. The success I told you about, that's the only success so far, and that's been LISTA, at least a successful trial leading to an FDA approval. And then I say the endpoint that I showed you was a big breakthrough because it worked. So the future, that was the past. There's an unprecedented amount of lupus clinical trials activity ongoing. But the challenges are there are not enough patients to fill these studies. The commitment that the company who developed Benlysta made to the FDA included about 5 what we call post-marketing trials, to do a study in blacks, to do a study in lupus kidney disease, to do a study in kids, and to do some other safety trials. And I think alone they need about 4,000 or 5,000 patients for their studies. There are about 12 to 13 other drugs being developed in lupus, and I think a lot of other companies who want to go forward in lupus. I don't know where all the patients will come from to develop these drugs. Already, we've gone worldwide. These studies are being done in every sector of the world. And trial design does remain challenging. So I mentioned before, even though we've been doing this for 18 years at least, and you think we would know how to design a trial, it still remains very challenging. A little easier for lupus kidney disease than it is for the extrarenal manifestations, but it's still very challenging for both. So what does this mean? Move over ACR20 and DOS. Those are instruments that we use in our rheumatoid arthritis studies. And if you go back about 10 to 15 years ago, we were pretty much at the same point in rheumatoid arthritis that we are in lupus. We were struggling with endpoints, we were studying with trial design. But what's happened since 1999 in rheumatoid arthritis, there have probably been about at least 12 different drugs approved for the treatment of rheumatoid arthritis. And they've really impacted the lives of patients with rheumatoid arthritis in a very favorable way. So I think we're just about 10 to 12 years behind our rheumatoid arthritis colleagues. And we did see a success last year with Benlysta, and I hope there are many more successes. So move over instruments for rheumatoid arthritis and make room for the instruments for lupus. So that's an update on lupus therapeutics, maybe a little more than you wanted to hear, but you got the gist of it. So thank you.